Hello, and welcome to Chapter 3 of our microbiology course. This chapter is going to be all about prokaryotes, which is going to include the domains bacteria and archaea. This is going to have everything to do with morphology, appendages, motility, various layers, reproduction, and specific types of archaea. So go ahead, get comfortable, and we will jump right into this chapter. When it comes to the difference of prokaryotes versus eukaryotes, there are some pretty big differences, but I want you to know these three. The first is how DNA is packaged in both types of cells. In prokaryotic cells, the DNA is going to be free inside of the cell, meaning that there is no nucleus that is going to contain that DNA. Eukaryotes, however, are going to have a nucleus that is going to house the DNA, and also how the DNA is going to be presented inside of that cell is important too. In bacteria, the DNA is going to be circular and there's going to be a single chromosome, where in eukaryotic cells, the DNA is going to be linear, and those chromosomes can come in a variety of different numbers. For example, in humans, we have 46 total chromosomes. The cell wall makeup is also going to be a particularly big difference. We are going to talk substantially more about bacterial cell walls and how they're made up of peptidoglycan. Meanwhile, the walls of archaea cells are going to be slightly different. So we are going to talk about that a little bit later, while eukaryotic cells are going to be made up of polysaccharides. So that is that big difference there. And then the internal structures within prokaryotes versus eukaryotes is also very different. The primary massive difference is that there is either organelles or not organelles. Eukaryotes will have organelles like the smooth and rough ER, the Golgi apparatus, the nucleus, mitochondria, all kinds of organelles. Prokaryotes do not have any organelles, and that's a very distinct difference. This image is not very different from what we saw in chapter 1 in looking at the various parts of bacteria cells. So this is another nice illustration showing things for like the pilus, which are those little projection of hair-like features. You have the capsule, which is going to be outside of the cell wall, not always included in bacteria, but something we are going to talk about. You can have a cell wall and plasma membrane. A lot of people use those terms synonymously, but they are separate structures. We have the nucleoid, which is going to be where DNA is stored. We'll talk about that more too. Inside of the cell, surrounding the DNA, is going to be cytoplasm. That is that gel found within the cell. You're also going to find ribosomes, which that is one thing that bacterial cells and eukaryotic cells share. Ribosomes are always associated with proteins, so kind of keep that in mind. We'll talk about that more later. And then you can also have on some bacterial cells a flagellum. That is that whip-like tail. We'll talk about that more too. Now, we were talking about the structures on the outside of the cell, but we also want to talk about the cell's shape or the morphology. When we look at this word morph before ology, because we know that ology means study of, morph is going to mean the shape of something or its shape. When we look at the different shapes of different bacteria, it is important to note that we are looking at a single cell. So the three shapes I really want you to pay attention to here are in the red boxes. We have coccus, bacillus, and spirillum. The other terms we are going to talk a little bit about later, but these are going to be the three primary ones that we want to talk about for now. Now, the thing that I want to get across here is, again, these are individual cell shapes. And if we talk about multiple cells living together, this is a colony. And colonies have a different kind of morphology that we'll be looking for and not going to be described as coccus or bacillus or spirillum. These colonies are going to, I want you to think of it more like it's a city with many individuals living inside of that city. So colonies are going to be made up of many, many cells. It's the individual cells that we care about, that morphology. Now, those three sets of morphological features that I mentioned on that last slide, coccus, bacillus, and spirillum, we are going to talk about them a little bit more in depth here, starting with coccus or cocci. So these are going to be spherical or round shaped cells that can be found in a few different flavors. They can be single, they can be in pairs, so diplococcus. You can have tetrads, which is groups of four, or a sarcina, which is going to be essentially kind of like a group of 8 that create a cube, or something like a staphylococcus, which is more like a cluster. And that term should sound kind of familiar because there is a very common bacteria that we know that live on the skin, Staphylococcus aureus, and in the wrong strain, these guys cause MRSA in hospitals. Moving on to bacillus or bacilli, depending on if you're saying the singular or the plural. These guys are going to be rod-shaped bacterial cells that have a slight elongation. So they're quite literally like a little cylinder, if you want to think of it that way. Again, these guys come in a few different flavors. You can have a single bacillus. You can have a diplobacillus. Those are pairs. And then they can also come in strains, like a streptobacillus chain. These are kind of interesting in that an example, one that we have talked about already quite a few times, is E. coli. E. coli is a rod-shaped bacterium. Now, moving on to the spiral-shaped bacteria. These are kind of neat because of the way that the shape is, and also due to its shape, it means that it has quite a bit of motility, which also means that these guys are usually found hanging out alone. They are not usually found in groups or pairs or anything like that. So there are two flavors of these spiral shaped bacteria, You have spirochete, which is going to be more of a corkscrew type shape. What's kind of neat, the example I have for this guy is actually syphilis, the sexually transmitted disease caused by Treponema pallidum. That is the bacteria. And then we also have Vibrio, which is going to be a curved comma-shaped bacteria with a single twist. So these guys are still kind of a spiral, but again, they are called Vibrio. Now, a structure that you want to look out for when it comes to looking under the microscope, you'll always be looking for what is the shape and potentially color. We'll talk about that soon. But you also want to be looking out for things that are filamentous or thread-like. These guys are multicellular, and they grow as branching filaments with sporangia. This is not something that we necessarily want to see in our labs. And the reason I'm bringing it up is if you do see it underneath the microscope, something like what we're seeing in those pictures, then you need to tell me, because what you're looking at is mold and not a special new kind of bacteria. So we do not want to be working with mold due to the fact that they can become airborne. So if we start seeing that underneath the microscope, you guys need to like report it to me very quickly, and then we will dispose of it. The image you see here is a handful of colonies growing on a plate of agar. Now, you can grow bacteria in like 100 different ways in the lab. This is just one of them, and it's a great way to be able to look at and count colonies, as well as maybe looking at different types of bacteria growing within this single area. Now you can see those really bright yellow dots, as well as a handful of more white dots, as well as some more faded, larger splotches. So the individual little dots, those are individual colonies. And remember that a colony is going to be made up of many cells, in fact, thousands of cells. They're like a city. So kind of keep that in mind, that when we talk about colonies of bacteria, this is what we're talking about. So the structure of bacterial cells can be laid out in three primary areas. The first one being external, and where you can see the pink surrounding the outside of that cell. That is where we're going to be talking about appendages, much like I mentioned before, as well as surface layers, which we will also talk about too. That second feature is going to be the boundary. You can think of this part of the cell as being like the skin of the cell, and the primary part of this is going to be the cytoplasmic membrane. This is also synonymous with the term plasma membrane, probably something that you've already heard of, but just so you know, if you want to write it down, cytoplasmic membrane is also synonymous with plasma membrane. The most internal layers and internal structures are all laid out there, but the only ones that I really super duper care that you know are cytoplasm, ribosomes, and then the nucleoid or chromosome. We will talk about plasmids and endospores though too, later on as we go through these parts of the bacterial cell. So let's move into talking about them independently, starting with the external structures on the bacterial cell. All right, so starting with appendages. These guys are going to be on the outside of the cell. They are that external part of the bacterial cell. And we can lay these two major groups of appendages out based on their function. So the first one is going to be motility, and then the second is going to be for attachment. We are going to talk about these various structures that are found within these two functions. So let's go ahead and start with motility and start with flagella. So as I stated before, not all bacteria are going to have a flagellum or flagella, because sometimes they can have more than one. Kind of have the image of a sperm cell in your mind. We know what a flagellum looks like, but again, bacteria can be a little bit odd and there can be more than one. So the main function of a flagellum is to be used for motility. It's for self-propulsion, getting from point A to point B, typically in an aqueous environment where that bacteria lives. The flagellum is going to be made up of three different parts, and I do want to cover those three parts individually. So the purpose and how a flagellum is going to work is not dissimilar to like the blades on a helicopter. It's going to rotate to propel that cell through its environment. So let's talk about those three parts that allow it to do that. The first part is going to be the filament. This is made up of flagellin protein. I do want you to know that. So, gentle reminder, everything kind of comes back to protein. This is just the start of everything seeming like it's connected to protein. The second part is going to be the hook. This is going to be a curved connector piece, and it's going to act very much like a rudder because it determines the direction that the bacteria is going to be moving. The last part of the flagella is going to be the basal body. These guys anchor the flagellum to the cell so that the cell doesn't just fly right off the flagellum. It's going to, again, anchor the flagellum to the cell, and it's also going to help in making sure that that tail or flagella can spin or rotate. Now, there are only 4 types of flagellar arrangements, I want you to know. There are more than these 4, but I only want you to know these 4. They are the 4 most common types of arrangements. And the first one is going to be peritrichous. Peritrichous? We're going to go with peritrichous, but you can say it however you'd like, as long as you remember it. Peri, if you remember from A&P, is going to mean around, and these guys have flagellum coming off of many parts of the cell. This is used for agility. The next one I want you to know is lophotrichous, and these guys are going to have the flagellum coming off of just one end of the cell. Lopho means tuft. So if you want to think of it as like a little tuft of tails or a tuft of hair, that's fine. Both of these that I just mentioned have a lot of ability to move quickly. They have speed on their side, but the peritrichous is going to have agility. The amphitricious bacteria have 2 opposite side flagellum, and what this is going to do is allow the cell to change direction very quickly. And then if you need to know, amphi just means opposite. It's just describing that there are tails on either side. They're just opposite sides. And then our last one is kind of the classic flagellar arrangement look, like a sperm cell. These are monotricious. Mono meaning one. So again, you can kind of imagine in your head a sperm cell if you want to, but again, keep in mind that sperm, as we know it, is going to be eukaryotic cells, and we are talking about bacteria here. These are some real-world examples and pictures of the various flagellar arrangements. In the first one, you see that monotricious tail, just a singular tail. Just underneath it is the lophotricious, where you're going to have tufts of the tails, but they're coming off at either end, not to be confused with the top right image, indicated as B, as amphitricious, where they are on opposite sides, but it's a single tail on those opposite sides, compared to the image that we're seeing where we have multiple tails coming out on both ends. It's still not the same thing. And then we have our peritrichous there at the bottom right, looking terrifying and very alien-like, all the tails surrounding and coming off of all the sides of the cell. Again, that is used for agility. Now, before we move on from these flagellum and the fact that it causes the cell to exhibit motility, which is really important. Not all bacteria are motile. That's a very important thing to make a note of. So most move through the use of flagella in a series of movements called tumble runs, What a tumble run is it's sort of like a flopping in one spot and then shooting off like a rocket in a direction. There are two types of movement without flagella that I do want you to know. So these are bacteria that don't have a flagella, but they still move. This is gliding motility. You can think of it like how slugs or snails skate along on a surface using the slime or mucus that they secrete. So these bacteria use the same mode. They secrete a slime and then they move over the slime. Then we have this one, and I'm probably going to butcher how to say it, but bear with me. It is spirochetes. It sounds French because it is, and it sounds a lot like a move a ballerina would make because that's precisely what it's based off of. So it's a twirling of the cell that pushes it along a surface, and it is caused by periplasmic flagella called axial filaments. So this is a very cute move, and we'll talk about it a little bit more on the next slide. But it's a twirling move that allows the cell to twirl away like a little microscopic ballerina. Now, there is another type of motion or movement, but make sure you note this, it is not motility. The cell itself is not doing the moving, it is being moved. Let me say that again. The cell itself is not doing the moving, it is being moved. What we call this is Brownian motion. It is this concept that everything is made up of atoms, if you remember from physics. And atoms are always moving or shifting and bouncing off of one another. So prokaryotic cells are so gosh darn small that atoms will bump into the prokaryotic cells and cause them to move. Again, this is not the cell having motility. It is the cell being pushed or moved by atoms. So this is Brownian motion. So if I ask you on the exam, which one of these is not motility, you're going to choose Brownian motion because it is not an example of motility. Okay? And if you have any questions on that, you are always welcome to email me. So moving on, still talking about motility, we have this term taxis, and taxis is going to be directed movement, and this is going to be caused from those tumble runs. Remember, the tumble runs are the cell flopping and then shooting off in a direction. So we have two types of taxis that you need to know. You have chemotaxis, which is going to be movement or those tumble runs due to chemicals. So either moving towards a chemical or away from a chemical, depending on what it is. Because if we're moving towards a chemical, it might be that our bacteria is looking for a food source. If we're moving away from a chemical, it might be to avoid antibiotics and thus being killed. Phototaxis is the other type of tumble run, and it is due to light. So you can have positive, where the bacterial cells are going to move towards the light, or negative, where it moves away from the light. An example of phototaxis is that spirochetes. This is going to be where those corkscrew little guys are going to spiral away from light. So a lot of the times you guys will not see those little fellas underneath the microscope because they are absolutely booking it when you turn on the microscope and turn on that light underneath them. So this is a nice illustration of a tumble run in a bacterium. You're going to notice in the gradient of attractant concentration that our little cell is moving in the direction of something. So it's going from that light area to that more reddish area. He has a direction he wants to move, and it might seem that he's not moving in the best way because there's a direct path from point A to point B, but he's going A to B to C to D to E to F to G to H. So why do they do that? Why can't he just, go from point A to point B there at the end? Well, the indirect pattern of his path is because that these guys are just simple little critters. They don't have eyes, and really what they have to do is they have to sense and feel their environment in order to get to where they want to be going. So again, usually this is due to either being drawn to or away from light, or looking for food, or avoiding antibiotics. So those are going to be the big reasons that you're going to have a bacteria moving, and they can't really do it all the best. And that's okay. They're just simple little critters. Moving on to another appendage, this is fimbriae. Fimbriae are going to be hollow tubes, also made of protein, called pillin. And what these guys do is help the cell attach to surfaces. So if you look at that illustration, we can see the fimbriae coming off of the outside of the cell, as if the cell were like a sausage that got rolled around in a hair salon. They're just little hairs coming off of that fella. Now, the thing that's important to note about fimbriae is these guys can cause something called biofilms. We are going to talk about biofilms later in this chapter, but for now, just make a note that these fimbriae are going to help the cell attach to surfaces, which can cause biofilms. The next external appendage is going to be a pilus or a pili, or pili, however you want to say it. And these guys are important because they're going to allow the cells to transfer DNA from one cell to another. Now, an important thing to note is that prokaryotic cells are going to be asexual, meaning they create a copy or clone of themselves during reproduction. But that doesn't mean that they don't exchange information with other cells. And we will talk about this more later, but this type of exchange is called conjugation. Sometimes these guys are called a sex pilus. We'll talk about that more again later in this chapter when we talk about conjugation. Now, the last appendage that we have is going to be the nanotubes or nanowires. These are really recently discovered, so we're still learning their function, what they're used for, but what we can get thus far is, one, they're made of cytoplasmic membrane from inside of the cell. They're used as channels from one bacterium to the next in order to transfer things like amino acids, which amino acids for bacteria are going to be like a form of food. And then also for the potential purpose to harvest energy by moving electrons around from the environment. So again, this is still relatively new information, but hey, kind of cool. We're living in a really neat time where we're able to actually study bacteria and archaea, and these are the things that come up. So we're still talking about the external parts of the bacteria, and we are going to move into the S layers, which you can also think of as armor for our lovely little cells. So these are going to be single layers of thousands of copies of a single protein that are linked together, and I want you to have an image in your head of essentially chainmail. So the purpose of this S layer armor is going to prevent drying of the cell as well as prevent injury. This can also cause the bacteria to be particularly difficult to kill, which is not a good thing. An example that you can think of is C. diff, which we will talk about more later in this chapter, or both in this chapter and also in the semester. But C. diff is a particularly difficult bacteria to kill that you see commonly in hospitals. And another example is anthrax. The other surface layer that we find on the external part of the bacteria is going to be glycocalyx, which we can also call the capsule. These guys are going to be slimy or gummy outer layers, and not all bacteria have them. Some bacteria do. They are made of polysaccharides or glycoproteins, and what this slimy, gummy secretion on the outside of the cell is going to do is help the bacteria from drying out, as well as can be used for attachment. And these guys are also going to aid in causing diseases and forming, I told you we'd talk about it, biofilms. So I'll have a couple examples of some biofilms here coming up. In this image, especially in the purple zoomed in portion, we can see the glycocalyx or the capsule on the outside of those rod-shaped cells. So it's going to be that yellow halo surrounding the cell, which means that the capsule kind of in a way almost doubles the size of the cell itself, which is pretty crazy. And remember, these are going to be slimy, gross, gummy outermost layers. And again, not all cells secrete them, but some do. So more about the glycocalyx and its special functions. So it's going to increase the ability to cause disease. So glycocalyx, the slimy outside of the cell, that capsule will protect the cell itself against white blood cells, or phagocytes. And remember that that is going to be your body's natural defense that is going to have the ability to engulf and break down foreign cells or foreign debris. So with that outer capsule, the cell becomes very difficult to break down, which is kind of neat. It is also going to make it slippery so it can escape and then multiply within your body. Exciting, right? It's not a good thing. We are not thrilled about these capsules. And a couple examples that we see in bacteria that do have a glycocalyx is anthrax, meningitis, and pneumonia, just to name a few. Now, because the glycocalyx is sticky and slimy, it can form what is not dissimilar to like a big sticky net that causes other cells that are either similar to that bacteria or are that bacteria to stick together, as well as other maybe cell particles or debris or other bacteria can also get stuck in this big sticky net. And what we call this is a biofilm. So they can be pretty difficult to treat when it comes to antibiotics, because the outer surface of the biofilm might be killed, but the inside could survive. So antibiotics may only scratch the surface, literally scratch the surface, and then deeper in the biofilm, you may still have bacteria surviving, thriving, and growing that you can't get to with that antibiotic. So some everyday examples that might make you a little squeamish of glycocalyx or biofilms are plaque on your teeth, as well as that pink film on your shower curtains or in your bathtub or kind of around your shower stall, as well as within your pet bowls. And even if you have free flowing water, like if you have like a little fountain for your pet, biofilms can still grow in there too. So it's that slimy inner coating. So if you go home and you touch your pet's water bowl and you're like, ugh, gross, slimy, that's because it's a biofilm in there and you should clean it. Water bottles can also build biofilms, and especially your straws. So make sure you're either replacing them or cleaning them particularly good. Very important to do. Some medical implications is we can see these biofilms causing problems with various devices used in medicine. So plastic catheters, intrauterine devices, and metal pacemakers can also build up biofilms inside of them. And as you can imagine, that can cause a bevy of problems for that patient. Now, as I said before, they act somewhat like a net, these biofilms do, and it can catch bacteria, all kinds of microbes, as well as debris within that biofilm. And again, this makes it very tricky to treat with antibiotics. So if you look at that image on the far right, upper right, you can see that the outside of that biofilm could get treated, that's fine, but there is an entire center that might not get reached by antibiotics, which can cause a problem in and of itself. So big sticky net. So we are now moving into the boundary of the bacterial cell to talk about the cell envelope. Now, there are three possible layers that we care about in this section: the outer membrane, peptidoglycan, and the cytoplasmic cell membrane. Now, what's important to note about these three possible layers is that not all bacteria or archaea have them. So when it comes to the outer membrane, we're talking about gram-negative bacteria, which is probably a term you've heard of. We are going to talk about the differences between gram-positive and negative here soon. So just kind of put a pin in it for the moment and just make a note, these three possible layers and who has it. The second possible layer is peptidoglycan. That is going to be our gram-positive and gram-negative bacteria. And then our cytoplasmic or cell membrane, all bacteria and archaea are going to have this structure. So let's talk about the purpose of these three possible layers. Before, we mentioned that the boundary of the bacterial cell is going to be like a skin. So I do want you to think of these as skin, but it's going to be like different types of skin for the bacteria or the archaea. So the outer membrane, which is going to be that gram-negative bacteria only, is going to be composed of a lipid bilayer containing lipopolysaccharide, or an LPS. This is going to be important later, and we'll talk about it soon, but just make a note that that is what is going to be inside of that outer membrane. From there, we have peptidoglycan, which can be a possible outer covering of our bacteria, specifically for gram-positive and negative. So we are excluding archaea here. So peptidoglycan is going to give the cell's outside shape and rigidity. It's going to prevent the cell from rupturing, which we're going to call lysing. Okay, that is that term there, lysing. It has a long sugar chain that cross-links by smaller proteins, So everything comes back to proteins, guys. I told you. And then our third possible structure, which is going to be found on all bacteria and archaea, is cytoplasmic cell membranes. So these guys are going to regulate what comes in and out of the cell because it is semi-permeable, and it's going to have that lipid bilayer. We actually talked about that in chapter 1 when we were talking about phospholipids. So if you need to go back and check that out again, that was in chapter 1. So I do have this lovely image of a gram-positive and gram-negative cell layers cross-section. You can see on the gram-positive that it contains the peptidoglycan layer, and it's particularly thick. This is kind of an important thing to make a note of, but it's just a nice illustration. We are going to talk about this more. And the other thing I want to point out here is that purpley layer at the top of the gram-negative protein. That's going to be the lipopolysaccharide layer. So our kind of two big important things is that very thick peptidoglycan layer in the gram-positive and the presence of the lipopolysaccharide layer on the gram-negative. Okay, so scooting right along. So a little more in depth about that outer membrane. Remember that this is going to be only found in our gram-negative bacteria. And the reason why this is an important feature to make a note of is this lipid A endotoxin. And what I want to point out about it is that the fact that gram-negative bacteria have it means that it's dangerous to us if we get an infection. So that endotoxin, that lipid A, is going to be built into the cell wall. And if that cell should die due to like maybe a treatment of an antibacterial or something like that, what we will see is that the cell has a little bit of a hateful streak in it, and if it's going to go down, it's going to take the person who's infected with them with the bacteria. So if we treat that bacteria with an antibacterial, the bacteria dies. That outer membrane that is full of that lipid A endotoxin is going to be released, and that's not a good thing. We don't want this, and what it can cause is even if the bacteria is dead, that endotoxin can kill your patient or make them substantially worse. So that's not a good time, not a thing we want. So kind of keep that in mind. Moving on to the peptidoglycan layer, remember in gram-positive cells, this is going to be particularly thick. In gram-negative, not so much. So what this is made up of is murin, or chains of disaccharides. So we have sugar chains and protein chains, the protein chains being a little bit smaller. And what this creates is something kind of like a chain link fence that helps as a barrier to solutes in and out of the cell. So if there's a particularly low salt content outside of the cell, water is going to want to move into the cell, and this is not a good thing. So the cell is going to use this peptidoglycan layer to prevent being flooded with a solute, which is not what we want. So for the most part, it does pretty well, but it's not going to stop an absolute flood. That is not going to help. So something you have heard of probably before, which is gram staining. This is going to be the process by which we stain bacteria to figure out if it is positive or negative. We are going to do this in lab a few times. in order to get you used to the process. So it's using crystal violet, Graham's iodine, alcohol, and safranin. So this is going to be kind of an interesting process that you are going to need to essentially perfect in this class, because it's very important to identify what kind of bacteria is causing an infection. Remember that if it is that gram-negative bacteria, it's going to contain those lipid A endotoxins, and you need to know that before you treat a patient, right? So gram-positive bacteria is going to stain purple due to that really thick layer of peptidoglycan. And gram-negative is going to stain red due to a thinner layer of peptidoglycan. And that is what we're going to see, is that gram-positive bacteria is going to be particularly violet in color, and gram-negative has a tendency to be reddish pink when we look at it. So let's take a look at an illustration to get a better visual of what those layers look like in our different types of bacteria. So on the left, let's start with gram-negative bacteria. You have that outermost layer, which is the outer membrane. Then you have a thin layer of peptidoglycan, and then you have the cytoplasmic membrane. You also have the periplasm, which is going to be a fluid-filled place between the cytoplasmic and outer membrane, so make sure you make a note of those guys. Remember that gram-negative cells are going to stain pink, so I would maybe write that in here somewhere, that these guys are going to stain pink, and it's because of that thinner layer of peptidoglycan. On the gram-positive side, so on the right side here, we have that really thick layer of peptidoglycan and no outer layer of that membrane. Then we have the cytoplasmic membrane, and for whatever reason, I don't know if you guys can see it as of the moment, I cannot, but there should be little hair-like structures coming off of this guy called tachycoic acid. So these guys are going to be hair-like structures that are used to help identify species, and it also regulates ion movement, and that's going to be something that is specific to our gram-positive cells. Remember that these guys are going to stain purple, and it's because that peptidoglycan layer that is so thick is going to soak up the stain. And, you know, gram-positive has a lot of that particular layer, so it soaks it up a ton. There's also no outer layer, that outer membrane, so the stain can access the peptidoglycan layer substantially better. We are going to talk a little bit later about some other important features of these cells based on these structures. So we are going to see these images again. I just want to point out what the title says here, and when we look at this at the end of the chapter, that title will change. Now, in talking about our cell wall within our prokaryotic cells, we do need to talk about if they're not gram-positive or not gram-negative, then what are they? Well, they could be mycoplasmas, which are certain types of bacteria that don't have peptidoglycan. So this is a little bit funky, a little bit weird. And what this means is that that bacteria does not fall into positive or negative as far as being gram-positive or gram-negative. These guys are also very fragile as far as bacteria go, which is not a great thing for the bacteria, but it is better for us in being able to treat it. And then our last type of prokaryotic cell wall is going to fall within our Archaea domain, where we have essentially a fake peptidoglycan layer. So it's called pseudomurin, pseudo meaning fake, and then that murin referring to the peptidoglycan layer. I do want you to know all the differences and be able to compare these 4 variations in the cell wall for these different kinds of cells. I'm not going to go over all the parts of this slide. It's mainly to help you in that it lays out all of the different structures that we have talked about side by side as far as gram-positive versus negative. So this is for you guys. Do be able to compare the two, but this is a slide that you can use to study from. All right, so we are moving to the internal structures of bacterial cells. So this is that last part of the structures. So we want to talk about the cytoplasm first. The cytoplasm is going to be that cell gel, I like to think of it as, that is going to be found inside of the cell. Eukaryotes also have cytoplasm, so this is something that both of those types of cells share, is cytoplasm. It is made mostly of water, about 70 to 80%, and it's going to be a solvent for the cell, contents like sugar, amino acids, and salts. We are going to have another term here, cytosol, which is going to refer to just the fluid of the cytoplasm. And possible things that you can find within the cytoplasm of bacterial cells include the chromosome as well as plasmids. We'll talk about plasmids here when we get into binary fission, which is how prokaryotic cells reproduce. Ribosomes, remember, they have everything to do with proteins. Inclusion bodies, we'll talk about them soon. Microcompartments, similar to inclusion bodies, again, we'll talk about that. Cytoskeleton, which we will not talk about, and endospores. All right, guys, so last slide of the first part of chapter 3. This is going to be talking about bacterial chromosomes. So we are still on those internal structures of bacteria. We've got our single circular chromosome, so it makes kind of like a, if you want to think of it like a rubber band of DNA, because a rubber band is going to be connected. It's a hoop, if you want to think of it like that. Remember that the big difference between prokaryotes and eukaryotes does have a lot to do with the DNA. Bacterial chromosomes are going to be that circular single loop of DNA, while eukaryotic cells are going to have multiple linear chromosomes, also found in the nucleus. Meanwhile, bacteria are going to have that loop of DNA, that chromosome, within the cell, but not within a nucleus. It is free within the cytoplasm, but it does have a special spot. So this special area is known as the nucleoid. And the nucleoid is, again, kind of just this special designated area within the bacterial cell that the chromosome will reside. So it's not just free floating around within the cell willy-nilly wherever it wants to go. It does have a special designated area. And with that, guys, that is the end of chapter 3, part 1. Stay tuned for the second part where we talk about prokaryotic reproduction, which we call binary fission. So how do prokaryotes make a new cell? I hope you enjoyed this lecture, and I'll talk to you next time.